Today is our second lecture of the course Neurophysiology and Cognitive Science. As you remember, this is our schedule, so we will have the lecture on the next week, and we are going to have a lecture or maybe some practical lesson in the end of the March. Today we are going to talk about basic neurophysiology, so some information about neurons and synapses. Then we will have basic methods of studying the brain, the basics of neuroanatomy of the brain, and 4 lessons with the focus on cognitive sciences and higher brain functions. If you forgot our channels of communication, here they are. Don't miss entering the Google Classroom because after this lecture you will have a test in Google Classroom. So this is how the class looks like, and in the classwork section, I suppose you'll have the tests, and the first you'll have only one test, then the second one, and so on. On the previous lesson, I told you that there are some books on Google Drive you can use if you want to know more about neuroscience, and this one, The Fundamentals of Cognitive Neuroscience, a Beginner's Guide, I suppose the most integral, and if you are going to read some literature, you should start with this book, and you can read some additional materials if you want in this book. So today our topic is the basics of neurophysiology, and there will be 4 parts. The first part is the story of how we know that there are neurons in the brain and how we learned to distinguish them. The second part is about the excitation of neural cells, how it works. And the third part is about channels and other important membrane molecules that you can find in the neuron. And the last part, a few words about some system principles in the brain. So in this part of the lecture, I want to focus on what I have called the cellular principle of the neural system. So we called cellular as opposed to the system principles and functions of different parts of the brain, which will be discussed later. So cellular principles are those principles that are closest to direct physical measurements. So they usually are not connected with some behavioral tasks. So let's start. Until the 18th century, there wasn't cellular brain science at all. So it wasn't clear what transmits the signal from the brain to the body. Although neurology has already received some development, so simple brain surgeries have been performed since the days of ancient Egypt. Anatomists learned to isolate different parts of the brain but it was very difficult. Do you know why? Why was it difficult to isolate, to distinguish different parts of the brain from each other?
Maybe because they look alike.
Of course, they look alike, but
because it's more complicated to distinguish.
Because if you get the brain out of the skull, the brain looks like a fat or a water, so it spreads. And now we have a lot of methods how to make the brain solid, and it's much more easier to isolate different parts of the solid brain that's of fat or water brain. Okay, is it clear?
That means a brain is a liquid or semi-liquid.
I mean that there are no bones in the brain, and all neurons are they like a liquid fat. So when you put the brain on the table, it do something like, so it can't get a form.
Okay, I get it. Thank you.
Okay. And in my opinion, the history of studying the mechanisms of the brain begins from the moment when in 1791, Luigi Galvani published his work on electrical stimulation of the frog nerve. So until this moment, the substrate for the transferring of information in neurosystem was unknown. So after the work of Galvani, it became clear that if exposure to electricity can get in motion parts of the body, then perhaps electricity transmits information in the whole organisms in a natural form. By the end of the 19th century, optical methods and chemical techniques like fixation and staining had reached a sufficient degree so that one could try to see the particles of the brain. And it became clear that the transmission of information in neurosystem has chemical and physical components. And there were significant advances in the field of reception, like vision and hearing. Nevertheless, there was a cellular network dualism. So it was unclear. So it was clear that there are cells in the brain, and it was clear that some nerves transmit electricity, but no one really understood if these things are connected. So do cells transmit electricity, or there is some special syncytium, some special substance in the brain, some networks of wires that transfers electricity? So the answer wasn't clear. From the point of view of a modern person who knows that neurons, axons and dendrites are, and all this may sound crazy, but the fact is that in the 19th century, all information about cell tissue was very scattered. This is due to the fact that cells and fibers in the brain are packed very, very densely. If we took the modern picture of a section of neural tissue, like on this picture, which made with the help of an electron microscope, then on a quite small area, we will see cross sections of axons, connections of hundreds of different neurons. So there you can see some synapses, some parts of the somas, some parts of axons. So they are really mixed, and it's hard to distinguish separate cells and their forms and how are they connected. So therefore, at the beginning of the 19th century, they were able to discern various details of the neural tissue, but not whole individual neurons. And staining methods that worked well for histological studies of other organs, in which cells are more or less round and not mysteriously associated with movements, feelings, and thoughts, and stained all cells didn't work for understanding the structure of neural cells. So if you color all the cells in the brain, you can't distinguish individual neurons. What helped to solve this difficult question about the duality of the cellular network structure of the brain? Almost jokingly, I can say that this happens thanks to the crazy king of Bavaria. Bavaria is a part of modern Germany, and there was no Germany in 19th century, but there was Bavaria, Prussia, and so on. And King Otto was diagnosed with schizophrenia. The study of the cellular structures of the brain were greatly supported. And Otto attended doctor was von Goldman. And the neuroscience didn't help the crazy king in any way, but in the end, the king helped the neuroscience. So because of all these investigations, von Goldman had a student, Franz Nissl, who invented a method of staining nerve tissues. So here we almost can see some individual neurons. So not all of the cells are colored in the tissue. This method is still used today. Also, Franz Nissl observed facts of structures and morphological changes with some psychotic disorders, and he was a friend and co-author of Alzheimer. But a few years later, Italian physiologist and scientist Camillo Golgi invented a different method for staining neurons. Golgi's method is a silver staining technique that is used to visualize neurons tissue under light microscopy, and it was initially named the black reaction. So you see, because the neurons here are black. But now it's more known as a Golgi method. So yeah, you see, here we can distinguish different neurons, but in fact we can distinguish all the axons and dendrites. But on the Golgi method, we can distinguish axons and dendrites, so on. So we can now we can see the shape of the individual of the whole neuron. It doesn't look like some mixed tissue with all neurons. Golgi staining was used by Spanish neuroanatomist Santiago Ramon y Cajal to discover a number of novel facts about the organization of neural system, inspiring the birth of the neuron doctrine. So he was the first to give a clear description of neural structures, especially the cerebellar cortex, olfactory bulb, spinal cord, midbrain, thalamus, hippocampus, retina, and so on. At the same time, Golgi was an opponent of the cell theory, and Santiago Ramon y Cajal was its supporter. Interestingly, that in the Nobel speeches in 1906 by these people, they presented two opposite theories. So Santiago Ramon y Cajal supposed that there are neurons in the brain and they transmit the electricity, and Golgi thought that in the brain there is some special tissue like syncytium, and it transmits the electricity like wires. The next major milestone in understanding the work of neurons was the work of Sir Edgar Adrian. Sir Adrian's key result is the all or nothing principle. In 1928, he conducted his experiments to measure the electrical signal of neural cells caused by a physical stimulus. So in particular, he measured the signals of retinal cells and sensory signals caused by electrical stimulation of the skin of the frog. And in the second, the electrical stimulus was continuously decreasing while the amplitude of the impulses ultimately remained constant. But the frequency changed. You can see this on the picture on the right. So in fact, it was a measurement of the impulse of a single neuron. This conclusion was eventually made by Adrian, frankly speaking, even Julius Bernstein, so another neurophysiologist, also measured the electrical activity of neural fibers. But earlier, with a continuous change in neurostimulation, the response also changes continuously. So this was due to the fact that the number of involved axons in the neurofiber changed. And Adrian, he discovered, his discovery is very important because actually he was able to notice the quantization of this signal, so quants of neural fibers. And later in 1932, Adrian received the Nobel Prize for his work of neural function with Sherrington. Another important milestone in the field of chemical signal transmission was made by Otto Lovey in his famous experiment in 1921. Lovey himself writes that the idea of the experiment came to him in a dream, so when he slept. He cut out two beating frog hearts, one with a vagal nerve, which regulates the heart, and the other without a nerve. Both hearts were placed in the saline baths. First, Lovey stimulated the first heart with a nerve, and it began to beat more slowly. Then he took a solution from this heart and poured this solution over the second heart, and the second heart also began to beat more slowly. So this proved that there is some chemical substance that also transmits a neural signal. So after these experiments, by the 1921, the neuroscientists already know that so there are distinct neural cells. They transmit electricity. So and also there are some chemical substances which also can transmit the neural signals. After the war, in 1949, the voltage clamp technique was introduced, which was later used in famous experiments with the giant squid axon in 1952 by Hodgkin and Huxley. So they freed the axon from the exoplasm, so it was really giant part of the neuron, and filled this axon with solutions with different ions. As a result, they were able to write the Hodgkin-Huxley equations. So this is a citation of their original work. The equations for the dependencies of ion currents of different channels on potentials and time. So the equations included the rate constants of the reaction of closing and opening channels. Thus, Hodgkin and Huxley introduced the concepts of channels that hadn't yet been discovered. So they just thought that there are some ion channels, but they couldn't find them. So they didn't have any methods to recognize these molecules. And of course, they also received the Nobel Prize for the discovery in 1963. The question of the presence of a channel in the membrane remained unclear. Models of artificial canals were built, but for a long time it wasn't possible to find natural channels. But in 1976, Erwin Neuer and Bert Sakmann developed the patch clamp technique. Prior to this, the activity of neurons was measured by electrodes, which destroyed the neurons or measured the potentials outside the neurons. So it wasn't possible to measure the current in a part of the membrane, in a small part of the membrane. And in the patch clamp technique, a hollow glass electrode was used, which was filled with a solution and had various modifications. So in the modification of whole patch clamp, the glass electrode broke through the cell membrane, and in the modification of the patch clamp attachment, it was sucked from the outside. And nevertheless, as a result, Neuer and Sakmann were able to measure a single channel, so a current through single channel. On the left side, you can see the picture of currents. There is also a kind of quantization. You can see current through one channel or through two channels, and so on. By the 1976, we also already know that there are different channels for different ions. And we know some equations how the signal is transmitted through the axons. And so measuring it's all the history information I wanted to tell you. And now let's talk about some principles. So the first one is neuron resting potential. So the first thing you should be aware of is the resting potential of the neuron. I think you remember that a neuron is a nerve cell and it has a membrane that is charged. That is, even at rest, a neuron has a potential difference inside and outside the membrane. And this potential is approximately equal to minus 70 millivolts. Let's see where it comes from. On this picture, there is potassium and sodium inside and outside the cell. Let's designate sodium with blue circles and potassium as red circles. We see that there is a lot of potassium inside the cell and a lot of sodium outside the cell. So here you can find the concentration of sodium, potassium. And the inside is separated from the outside by a membrane through which nothing passes. The resting potential is the potential at which no current flows through the membrane. It's negligible. Let's designate the concentration of these ions through natrium, potassium, sodium, potassium, and chlorine. Add the subscripts I for inside and O for extracellular outside concentration. So on the one hand, in a unit of time, so many ions in one unit of time, we have a current from inside to outside, like this, and from outside to inside, like this.
It's occur in the same time?
Sorry?
It's occur in the same time?
Yes.
So ions to get out and ions to get in at the same time?
Yes, that's right. P here is some constant of membrane transmission.
It's through a bond who maintains this, right? The sodium-potassium bond.
Sorry?
I was saying that there is a bond who's responsible to keep this potential, right? The sodium-potassium bond.
Yes, sir. But now we are not discussing the channels and pumps. So we just try to find the equation for resting potential.
Okay.
Using some physical ideas. Okay. So we can write the equation for current from inside to outside. Here you can see the equation. And we have a product on exponent from minus u divided kt, so it's kind of Boltzmann.
And what it means?
It means that if you have some potential, so
I mean, what does U refer to and KT refer to?
These ions, they have lower energy because, for example, if you have a field from this side to this side, the positively charged ions can transmit easily from the right to the left, and only only the ions with very high energy can transmit from the left side to the right side. Okay. So this is.
I think no. I was asking about what is this mean? What U symbol is referred to?
The U is the potential energy.
What do I mean by this product or by the current?
This equation, I mean, this equation, yeah.
I just want to calculate the current from inside to outside.
Yeah, and it depends on the energy. If we have a high energy, so the direction, it will be from outside to inside.
Yeah, the energy of the ions are connected with their temperature, according to physics. Okay.
Okay. Yeah.
And so if they have got high temperature, the ions have high energy, and they can go through the membrane instead of the voltage, instead of the field. So they charged, for example, if the ions charged positively and the field is from outside to inside, they can't go outside because of the field. But if they have got high energy because of their temperature, they can do that. And if we want to find how many neurons can transmit from inside to outside, we should make this product. And to calculate the current from outside to inside, we shouldn't do any product because there is a field.
Because it's not need energy to.
Yeah, they don't need energy.
Yeah.
Okay, and so what we have? We have current into the cell. We have current out of the cell, and in the resting state, they are equal. From this idea, we can find the Goldman equation. So we can find the resting voltage difference between inside and outside. And the first approximation, we take into account that pK is much larger than P sodium, and so we have this equation. And this is about from 50 to 100 millivolts, and we called it Nernst equation. So this is how you can find the difference in resting state for neural membrane. Now let's talk about neuron action potential. So an action potential occurs when the membrane potential of a specific cell location rapidly rises and falls. This depolarization causes adjacent location to similarly depolarize. So the depolarization transmits from one part of axon to another part and to another part and so on. Action potentials are generated by special types of voltage-gated ion channels embedded in a cell's plasma membrane. These channels are shut when the membrane potential is near the resting potential of the cell, but they rapidly begin to open if the membrane potential increases to a precisely defined threshold voltage, depolarizing the transmembrane potential. So when the channels open, they allow an inward flow of sodium ions, which changes the electrochemical gradient, which in turn produces a further rise in membrane potential towards zero. This then causes more channels to open, producing a great electric current across the cell membrane and so on. So here we can see the increase of membrane potential. And the process proceeds explosively until all of the available ion channels are open, resulting in a large upswing in the membrane potential. The rapid influx of sodium ions caused the polarity of the plasma membrane to reverse, and the ion channels then rapidly inactivate. As the sodium channels close, sodium ions can no longer enter the neuron, and they are then actively transported back out of the plasma membrane. Potassium channels are then activated, and there is an outward current of potassium ions, returning the electrochemical gradient to the resting state. And after an action potential has occurred, there is a transient negative shift called an afterpolarization. If you know physics a little bit, maybe these graphs can be useful for you. So let's look at the graphs to understand the physical essence of this process. At the first graph, the dependence of the number of ion channels of sodium and potassium on the potential. The second graph shows the characteristics of current and voltage for sodium, potassium, and their sum. So the green line is their sum. The points of intersection of the total graph are the resting potential and the action potential. So there is an action potential. Such wave propagates along the axon during the action potential. And you have probably heard that there is a myelin sneath of some axons. And this is a liquid membrane formed by glial cells. Part of the axons of our body, or for example, the body of a giraffe or an elephant, are surrounded by such a shell. Ion channels are located between the intersections of the early, that is, in those areas where there is no myelin. So we haven't got ion channels under the myelin shell. And the field is well transmitted through these areas with myelin shell. So the speed of signal transmission, even along 10-meter axons surrounded by myelin, is quite high. So because we just need time to open and close the channels, but the field transmitted almost immediately through this part of the neurons. So if you want to find the speed for transmission the signal through such a long neuron, you just should sum up the length of the parts without myelin and to find the speed of transmission with this shortened axon. Now we have figured out that all signals transmission in neurons occur with the help of special transmembrane complexes, channels, and let's talk about them in more detail. So a cellular membrane possesses two key qualities. They're semi-permeable, so only certain materials may freely cross. Large and charged substances are typically blocked, and they are selective. So membrane protein may regulate the passage of material that cannot freely cross. There is a passive transport and active transport. And passive transport involves the movement of material along a concentration gradient, so from high concentration to low concentration. Because materials are moving down a concentration gradient, it doesn't require the expenditure of energy. So it doesn't require ATP hydrolysis. There are three main types of passive transport: simple diffusion, movement of small and lipophilic molecules, osmosis, movement of water molecules, and facilitated diffusion, so movement of large or charged molecules via membrane potential, like ions and so on. And there is an active transport. Active transport involves the movement of materials against a concentration gradient, so from low concentration to higher concentration. Because materials are moving against the gradient, it requires the expenditure of energy. And there are two main types of active transport: primary or direct active transport. It involves the direct use of metabolic energy to mediate transport, and secondary or indirect active transport. it involves coupling of molecules with another moving along an electrochemical gradients. There are three major molecular complexes built into the neuron membrane that provide rest and action potential. Complex molecule is a potassium-sodium pump or potassium-sodium ATFase. This complex consumes cellular fuel, converts ATP molecules into ADP molecules. For such a transformation, 2 potassium ions are thrown into the cell and 3 sodium ions are removed from the cell, like on this picture. Thus, due to the pump, a negative current is constantly injected into the neuron. It's thanks to the potassium-sodium pump that the neuron restores the concentration of potassium and sodium after the neuron is excited. The next thing you should be aware of is how the synapse works. So the synapse is a connection of 2 neurons. On the right picture, you can see the synapse from electron microscopic picture, and the central picture is just a computational picture. So this is a modeled neuron with many synapses here. In fact, synapses can be chemical and electrical, but electrical ones are not very common. In electrical synapses, the field is transmitted directly. So electrical synapses are connected by special proteins called connexins, and they are also called gap junctions. As we already talked a little, there is a scheme of synapse. So there is a presynaptic part of the presynaptic cell and part of postsynaptic cell, and this is an axon terminal. There are some synaptic vesicles with neurotransmitter, and there are voltage-gated calcium channels in the presynaptic neuron. There is a synaptic lift, and there are some receptors for neurotransmitter or ligand-gated ion channels in postsynaptic cell. And when action potentials reaches the axon terminal, it depolarizes the membrane. The voltage-gated calcium channels are opened. The calcium flows in, and the calcium influx triggers synaptic vesicles to release neurotransmitters. Then neurotransmitter binds to these receptors on target cell, so this is in fact the transmission, electrochemical transmission of the signal in the synapse. I just want you to remind about the measured neurotransmitters and how do they affect the brain. Here you can see that 19% of synapses are glutamate, It's the measure excitatory neurotransmitter in the brain, and the measure inhibitory transmitter is GABA. The funny thing is that even the glutamate, for glutamate, there are several different receptors in the post-synapse. For example, there are AMPA receptors and there are NMDA receptors, and both of them are receptors for glutamate. They also have different pharmacokinetics and different speed of response and the duration of activation. In addition, the receptors in addition, AMPA and NMDA receptors, both of them, they are for sodium, but only NMDA can transmit calcium, can pass calcium. So in general, synapses are not simple. So there are a lot of molecules in synapses, membrane proteins, and so on. So there is just a simple diagram, but in fact there are many more. For example, there are some special molecules like neuroligin and neurorexin for connection different types of synapses. Another important thing to remember is that there are two types of receptors, ionotropic and metabotropic. In fact, the ionotropic receptors is a channel that can open in response to the attachment of a ligand, when the metabotropic receptor is a G protein coupled receptor that triggers a cascade of internal processes in a neuron. So the ionotropic receptors are faster. They triggers by action potentials, and they can be made up from different molecules. So there are more than one molecule in one ionotropic receptor. And metabotropic receptors, they are slower. They can amplify or dampen signals. They also can trigger different postsynaptic events, and they are monomers, which means that there is only one molecule in this receptor. And metabotropic receptor mainly respond to modulators like dopamine, serotonin, and norepinephrine. And ionotropic receptors, they are usually for key neurotransmitters like GABA, glycine, and glutamate. So you see here, ionotropic receptors are usually connected with glutamate and GABA, and metabotropic receptors, they are connected with other neurotransmitters or neuromodulators. And finally, a few words about system principles. So as we decided earlier, one neuron transmits a signal to another, but this is usually not enough for second neuron to generate an action potential as well. And this usually requires the second neuron to receive many influences from other neurons which are summed up. On the basis of this principle, the first model of an artificial neuron was built. It was in 1943, and the inventors of the first artificial neurons were McCulloch and Pitts. Neurons in artificial neural networks still work almost like that. So there is they sum up the signals, the weighted signals from the previous neurons, and then if this sum is more than some threshold, then we have 1 for this neuron, and if the sum is lower than the threshold, we have 0 for this neuron, for almost all or nothing principle. But for real neurons, we haven't got just ones and zeros like for artificial neurons. So we have, in real life, we have spiking neurons. So they can do the action potential or the spikes in every moment. And so it's kind of continuous model. Now we are not sure how do the neurons transmit the signals in general. So for some neurons, we know that the frequency plays the major role, and for other neurons, we know that the time of the spike plays the major role. So there are different spiking codes for different neurons. For example, in some receptive systems and in the center of the brain. So in the real brain, the neural code is more complex than just ones and zeros, and neuron can encode frequency, and also its potential can be taken into account, because sometimes we haven't got a spike, but we have some differences in membrane potential, and it also can play a role. So things are more complicated, but we will talk about neuron system in next lectures. So that's all for today. You can ask questions if you have, or see you after a week.